So, have you ever wondered how doctors and scientists can look at a tiny tissue sample and know exactly what's going wrong inside a patient's cells? Well, welcome to this explainer. Today, we're going to unlock that exact mystery. We're diving into Professor Sameh E. Hassanein's fascinating lecture on how healthcare data science allows us to act as biological detectives. And our main tool for this investigation? A revolutionary technique called RNA sequencing, or RNA-seq for short.
Here's a quick roadmap of what we'll be covering today. First, why RNA sequencing actually matters. Then, how we gather the genetic evidence, the file types we use like FASTA and FASTQ, how we reconstruct the genetic puzzle, the difference between discovery and comparison, and finally, how we filter out statistical noise. Let's get to it.
Starting with Section 1, why RNA sequencing matters, which really is the ultimate biological detective work. To really grasp this, we first need to understand what RNA actually is. Think of it this way. If your DNA is the massive, permanent reference library hidden deep inside your cells, RNA is like the temporary photocopies of specific pages that the cell is actively reading right now to build things and stay alive. RNA sequencing is simply the process of reading those active instructions to figure out what the cell is doing at any given moment. Professor Hassanein's lecture at Nile University shows us that mastering these computational workflows lets us peek right behind the biological curtain. By looking at these RNA messages, we can see exactly how a disease is behaving or how a new medical treatment is changing things in real time. But, as you might guess, doing this requires an absolute mountain of data. Moving on to Section 2, gathering the genetic evidence and the best practices for doing it right. So we can't just read an RNA molecule from start to finish like a normal book. The technology relies on a process called sequencing by synthesis. Imagine taking a massive library of instruction manuals and running them through a paper shredder. Those tiny shreds are called reads. We use a high-throughput platform like the Illumina HiSeq to scan those shreds. Now, our source notes some strict best practices here. You want a minimum read length of 50 base pairs. Think of a base pair as a single letter in the genetic alphabet, though 100 letters is much better. And we need a ton of these shreds. We're talking a read depth of 30 to 40 million shreds per sample. Plus, we need 3 or more biological replicates, which just means running the experiment on multiple different samples so we know our findings aren't just some random fluke. That brings us to Section 3, FASTA, FASTQ, and quality, where we start inspecting our clues. Before we can even think about taping our shredded manual back together, we have to store all these short genetic sequences in massive text files. In the data science world, you're going to hear about 2 main file types, FASTA and FASTQ. It's actually pretty simple. FASTA is just the raw text of the shredded book, a massive string of letters. But FASTQ is that exact same text plus a built-in confidence score for every single letter. It's literally like a spell checker telling you exactly how confident the sequencing machine was when it read that specific piece of code. And that quality value, it's absolutely vital for the next step. You see, machines aren't perfect. We have to do quality control, or QC, trimming. This means using software to chop off leftover chemical adapter sequences and any bad bases from the ends of our reads. Imagine cutting off the blank, frayed, or smudged edges of our shredded paper to get rid of the junk evidence. If a read ends up being too short after trimming, say under 20 base pairs, we just throw it in the digital trash. A fragment that tiny just doesn't give us enough context to figure out where it belongs, and it literally just bogs down the computer. Getting this trimming right improves absolutely everything we do downstream. Section 4, reconstructing the puzzle, which is all about aligning those reads. Okay, so our frayed edges are trimmed and our data is sparkling clean. But now we face a monumental computational challenge. How on earth do scientists tape 40 million shredded genetic pieces back together? How do we transition from a chaotic pile of data into a beautifully organized map that tells us the full story of the cell? Well, there are 2 main ways to solve this puzzle. If we already have a master copy of the instruction manual, like the standard human genome, we use what's called reference-based alignment, using software like the popular Tuxedo suite. It's exactly like putting together a jigsaw puzzle while looking at the picture on the box. But what if we're studying some totally new, unsequenced organism? We don't have a box cover. In that case, we build it completely from scratch using de novo assembly tools, like Trinity. We literally just look for pieces that happen to overlap and slowly build the picture completely blind. When we do have that master genome, scientists often rely on an alignment tool called Top Hat. Now, human genes are tricky. They aren't just solid blocks of text. The cell actually chops them up and splices them together in different ways, almost like a movie editor combining different scenes. Top Hat is a brilliant piece of software because it's uniquely splice-aware. It understands how these genetic sentences are stitched together, which lets it accurately map our tiny shreds right back to the master genome.
All of this computational heavy lifting is exactly why modern biology is, at its core, a data science. You need serious processing power and complex algorithms to take millions of puzzle pieces and figure out where they go. Our source notes that a good alignment should hit a 70 to 90% unique mapping rate. Basically, we want to find the exact, unique home for 70 to 90% of our shreds. Section 5, Discovery versus Comparison. This is where we analyze our output.
Once the manual is rebuilt, our detective work goes down one of two paths. First, there's discovery. This is all about exploring the unknown. We might find completely undiscovered genetic instructions or figure out exactly where a specific gene starts and ends. The second path is comparison. This is where we take samples from different conditions, like a healthy cell versus a cancer cell, or a patient before and after taking a drug. We compare them to hunt for the specific culprit causing a change. We're basically asking, did this new medicine turn the volume knob up or down on this specific gene?
And finally, Section 6, Filtering Noise from Reality and Establishing Statistical Certainty.
When we're comparing gene expression, we hit a huge hurdle, distinguishing real biological changes from simple measurement errors. Just because a computer says a gene's volume changed doesn't mean it actually did. We have to filter out what statisticians call Poisson variance or counting noise. This is just a fancy way of saying that when your sample sizes are tiny, random chance creates huge uncertainty. Think of it like this. If you're counting red cars on your street, and you see one today and two tomorrow, did the city's red car population actually double overnight? Probably not. It's just random chance.
Let's look at a concrete example. Say we're looking at gene B. We see one read in our control group and 2 reads in our experimental group. Now look at gene C. We see 100 reads in control and 200 reads in experimental. Mathematically, both show a 2 times difference, a doubling of expression. But jumping from 1 read to 2 reads is incredibly uncertain because of that Poisson counting noise. It's probably just a fluke. However, jumping from 100 to 200 reads, that is solid, undeniable evidence of a real change happening inside the cell.
And this is exactly why we need those massive read depths and biological replicates we talked about earlier. By making sure we have enough data, we can confidently separate technical noise, which is just a glitch or a slight measurement imprecision in our sequencing machines, from biological variance, which is a true, meaningful difference happening inside the actual patient.
As our source material brilliantly reminds us, we have to know the limits of the experiment. We can't just blindly trust the very first number a computer spits out. The hallmark of truly great data science is using multiple approaches, understanding the math behind the software, and knowing exactly where your tools excel and where they fall short.
So now that you've seen how we gather millions of genetic shreds, clean them up, reconstruct the puzzle, and use powerful statistics to separate fact from random noise, what other microscopic mysteries do you think we could solve next? The ability to read the live instructions of a cell is absolutely a game changer. It opens up an entire universe of possibilities for curing diseases and understanding life itself. Keep that curiosity alive, and thanks for joining me on this explainer.